Okay, so Monday, when you walk in, your responsibility is for you and your group to inoculate your knowns and unknowns on the media, get them in the incubator, and then you're done. And we'll look close on Wednesday and start collecting the first information about your unknown. Okay, so did y'all figure out who's doing what when you come in? Your group? Huh? Remember, each group was supposed to decide who was going to do what known organism before you left on Wednesday? You didn't do that. You should do that today. Okay, next material that is on Dropbox, the lecture and the PowerPoints are both titled Growth. We won't get through all of those for the exam, but I'll tell you what breaking point will be, but we'll get about halfway through. We'll talk about growth and how you measure growth, and then we'll stop at the point where we talk about some factors that affect growth. So that'll be the breaking point for the exam. We did update the Dropbox. If any of you want to do those Mastering Microbiology online quizzes for optional points, I told you, unless you had the e-text and you wanted to do some reading for those quizzes, you'd have to use my one copy. What I did do was I found instructor guides for all of the chapters, and they're a summary of the chapter, kind of section by section. So that's what's now been posted on Dropbox, and so you might be able to use those for the quizzes if you don't have the e-text and you don't have access to this particular book if you want to use those.
So how many optional points do you get? Like, is it based off the grade we get on the quiz, or how does that work?
You have to get 100% because you can
Okay.
retake it.
Okay.
Yeah, I think I put that in the instructions that I sent out about Mastering Microbiology. So you can retake it and it will tell you what the right answers are, but then it puts them in a different order.
Got it.
So. Anything else? Don't forget to do your absorbance of your unknown if you haven't done that. What I would recommend doing then is, once you know what the temperature optimum is, go back to your original culture I gave you and do a streak plate, incubated at that temperature, which you've now determined is your optimum. And then when you have isolated colonies on that streak plate, use an isolated colony to start new working in reserves. Because if I take a slant with just a whole bunch of growth on it and take some growth from that and just do a new working reserve, if it's contaminated, those working in reserves are going to be contaminated. But if you're working from isolated colonies, you know they're not contaminated. So if you can get in that habit of taking your reserve culture, streaking it out, and then starting new working in reserves from isolated colonies, you're really going to minimize your chances of contamination. Okay? Is there something flying around here? Little gnat. I think Jim fixed the whining faucet, so hopefully we won't have to deal with that anymore. Yes?
How long can you keep the streak plates in the incubator?
They shouldn't be in for more than 48 hours.
Okay.
Okay. So if you want to do it today, you can leave it in over the weekend. That's fine. Okay. Okay, I think we should be able to finish up the dreaded overview of Bergey's manual and kind of the taxonomy section. And my intention is hopefully we'll be able to start the next material on Tuesday, dealing with growth of microorganisms. So we're going to jump into volume 3 now, which are the low GC gram-positive bacteria. There is a little caveat here. Most of these organisms are gram-positive, but one of the 3 classes in the only phylum, there's a single phylum for Mycides, one of these 3 classes contain mycoplasms. And mycoplasms don't have a cell wall. And if you don't have a cell wall, will you stay gram-positive? No. If you lack a cell wall, you'll stay gram-negative, because there's nothing to retain that crystal violet iodine complex following the decolorization with the ethanol. So these all are low GC. Most of them are gram-positive without one particular exception. So let's take a look for just a second at the first class, the class Clostridia. And within that, there is a genus called Clostridium. And within that genus are two species that we will talk about in more detail later on. The genus Clostridium, or the species Botulinum, and the species Tetani.
When like cows first have their milk come out, it's called colostrum. Is there any relation? Is that like the first like?
No, unfortunately, no. Yeah. Same for humans. The first milk that's produced is called colostrum. No relationship. Good question. One of the things that characterizes members of this particular class is that these organisms are strict anaerobes. They are actually poisoned by the presence of oxygen. And when we talk about microbial metabolism, we'll give you an idea of what that poisoning entails and why oxygen is toxic. So you have to be able to grow these in special conditions that exclude the presence of oxygen. Naturally, where can you find them? Well, you can find them in mud layers where oxygen can't diffuse down through the mud. You can even find them in intestines because there can be fairly anaerobic conditions in part of your large intestine in particular. The microbial populations that are there metabolize the oxygen and use it up, and so they can create little anaerobic pockets. So it is possible to have anaerobes in the gut as well. What do you suppose Clostridium botulinum causes? Botulism poisoning. Okay. What about Clostridium tetani? Tetanus. Okay. Both of these diseases are the result of a toxin that the bacteria produce as a result of their metabolism. And we'll talk later on when we talk about toxins about the physiology involved and how these particular toxins actually cause disease. How can you minimize your chances of getting tetanus? Vaccination, okay. And you're not vaccinating against the causative agent, the bacterium. You're actually vaccinating against the toxin itself. Okay. The DPT inoculation you get for diphtheria, pertussis, and tetanus are all inactivated toxins that cause those particular diseases. Botulism, anybody know what the primary source of botulism poisoning is? These particular bacteria are very commonly found in the soil. If you do an anaerobic culture of soil, you'll get a lot of clostridial species that will grow. And so vegetables, particularly that touch the soil, get routinely contaminated with these particular bacteria, and these bacteria produce spores. The spores are resistant to boiling, and so if you eat some green beans, for example, that have been boiled but not autoclaved or pressure cooked, those spores can survive and germinate, and then they can produce the toxin and cause the disease. So a lot of times you'll get these through improperly canned food, things that haven't been sterilized the way they should have to kill those botulism spores. What's a relatively recent medical application of the botulism toxin? Botox, okay? These particular toxins tend to paralyze muscles. What Botox does in small doses is it paralyzes facial muscles that are responsible for wrinkles. Okay, actually contraction of those facial muscles causes the wrinkles. And so if you paralyze those, then it can relax those particular muscles and relieve the wrinkles. But the dosing obviously is very critical because in too high concentration, muscles that can be paralyzed by the botulism toxin include your diaphragm and your ribcage muscles, and so you can't breathe.
People who go through Botox build a resistance to botulism.
Good question, and I don't know for sure, but it would seem that that would be a likelihood because you probably would start producing antibodies against that. Good question. Second class within this particular volume 3 is this class Molecules. And within the class Molecules is the genus Mycoplasm. And we've already talked about mycoplasm in the context of this is an unusual bacterium that lacks a cell wall. Remember how they compensate for the lack of a rigid cell wall? Yeah, they have sterols in their cell membranes, which is unusual in prokaryotic cells. Usually those prokaryotic cells lack sterols in their membranes. Because these lack a cell wall, they have highly variable shapes depending upon environmental conditions. Do you remember what that terminology is that said that you can have a lot of different shapes? Pleomorphic, good. Okay. These organisms have a variety of different shapes depending upon different conditions. Oops, forgot to show you a couple slides. Here are some Clostridia bacteria that have invaded human tissue and produce gas as a result of their metabolism. And so there are gas-filled spaces that can develop in the tissue as a result of this clostridial infection. Okay, here's our mycoplasms. Okay, again, you've seen the slide before. Highly pleomorphic, different cell morphologies, even within the same population. Probably one of the contexts in which you've heard of mycoplasm before is that mycoplastic pneumonia sometimes is referred to as walking pneumonia. Have you ever had walking pneumonia before? Yeah, it's not pleasant. It's not pleasant. It is a respiratory disease. I think that it was originally called walking pneumonia because it was considered to be a little bit less severe form, and therefore you can still function. Whereas if you have a full-blown case of bacterial or viral pneumonia, you're probably out. But this one's kind of difficult to treat. There are only certain antibiotics that work against these mycoplasms. And sometimes it's quite hard to diagnose this particular situation. And then the third class within this volume 3 is the class Bacilli. And we're going to get into the class Bacilli in just a little bit more detail than we have the others. There are 3 genera that we're going to talk about in this class, and one is the genus Bacillus when you looked at morphological forms in the first lab, the slides labeled C for cocci and B bacillus. And so those particular bacteria were all in the genus Bacillus. And they're characterized by being gram-positive rods, and most of them will produce endospores. That's usually one of the defining characteristics. Clostridia are also gram-positive rods that can produce endospores. The big differentiation is that the Clostridium are anaerobic, and these are typically either aerobic or facultatively anaerobic. You know what that term facultative means? Facultative means you can do either or, basically. So you can respire in the presence of oxygen and do aerobic respiration, but then if there's no oxygen present, you can do anaerobic respiration. In these particular slides right here, you can actually see the endospores as these kind of refractory areas inside of the cells, because the endospores don't take up the stain readily unless you do something really severe to them, like heat them and steam them, which will drive the stain into the endospore. That's what we'll do when we do the endospore stain. A lot of common bacillus within soil populations, and some of these particular species produce antibiotics. Antibiotics are actually produced during the process of fermentation. What would be the common thought about why a particular bacterium, like a soil bacterium bacillus, might produce an antibiotic?
Kill everything that's competing with it.
Yeah, that's always been kind of the wisdom, is that it is kind of a chemical warfare that cells can wage against their competitors, and it then gives you an advantage. But I was just reading an article just recently now that's starting to question that, and they're kind of wondering if that's really the purpose of producing antibiotics. So the jury might still be out on that, but that's kind of the widely accepted idea right now. The causative agent of anthrax is caused by Bacillus anthracis. We talked about anthrax in the context of kind of the history of microbiology when we talked about the germ theory of disease. And Koch's postulates, remember he was working with anthrax and found those Bacillus bacterium in the blood of the cattle that were infected by anthrax. Fair number of you will have unknowns that will be in this particular genus. And this is a situation where identifying to the genus Bacillus is really pretty straightforward. Okay? If you're a gram-positive gram-produced endospores, very likely it's going to be Bacillus. But it gets to be a challenge sometimes in identifying the species. And that's where you get into some of the metabolic tests and some of the distinctions like location of endospores within the cell, size of the endospores. And so just because you have an easy time getting to genus doesn't mean necessarily it's going to be that easy to get to species as well. So we're going to be working with, throughout the course of the semester, Bacillus cereus, Bacillus subtilis, Bacillus megaterium. There's several different species of Bacillus that are pretty commonly used in the micro lab. Another genus within this class is the genus Staphylococcus and the genus Streptococcus. Now, this again points out kind of the new reality of classification based upon genetic composition. Because in the past, in the old editions of Berge's manual, these would have been in very, very different parts of the keys, and people had assumed that these had very, very different evolutionary histories. That's based upon the assumption that one being a cocci and the other being a bacillus, that dramatic difference in morphological form must have been indicative of difference in genetics. And as it turns out, not so much. Okay? So now in this new classification, they're both in the same class, bacilli. So you've seen some staph bacteria already in lab when we looked at the cocci. These clusters of cells, the irregular clusters is the Staphylococcus arrangement. If the cells are in chains instead, can I have one of those, please? Then they're called Streptococci. And this electron micrograph gives you a little bit better picture of what the individual cells look like. Sometimes in these clusters, it's hard to see individual cells, and it's pretty tough to measure them. Okay? I don't know if you remember or not, but these are really down in the kind of micrometer range, 1 ocular unit in diameter. And remember on your oil tower, the standardization is 1 micrometer per ocular. So those are really pretty tiny. One of the things that both of these genera are noted for are causing various diseases. These two particular type of bacteria are sometimes referred to as being pyogenic. Pyogenic literally means pus forming. What is pus? It's accumulation of dead red blood cells, white blood cells that have moved to that site of infection to try to kill the pathogen, and the pathogens themselves. And there's some fluids and things that result from inflammation. We'll talk about inflammation later on. but it's this kind of nasty conglomerate of dead tissue, white blood cells that have died trying to kill the pathogens and the pathogens themselves. Why are these organisms pus forming? Well, they're fairly notorious for producing a class of compounds called leukocytins. What does cytal mean? Homicidal, matricidal, fratricidal. To kill, right? Okay. And leuco refers to leukocytes or white blood cells. And so these are toxins that kind of target white blood cells and kill them. It's one of the defense mechanisms that these particular organisms have. Staph cause a number of skin infections. And if you want to get grossed out sometime, just go on the web and start looking up, staphylococcal skin infections, and you'll get all kinds of nasty images of different types of things that they can cause. Boils, carbuncles, furuncles, all these different nasty things that they cause. Here's a couple of examples of staphylococcal skin infections. Most acne is caused by staphylococcal infection of hair follicles. Impetigo's in there, yep. And then there's some other interesting diseases that staph can cause as well. And we'll get back to these in more detail when we talk about bacterial toxins. But there's a pretty bad toxin called toxic shock syndrome toxin. And that can be produced by staph. And then we have the issue of antibiotic-resistant strains of staph, like MRSA. And these toxins, if they become not just localized but systemic, can kill you. And that's particularly the case with toxic shock syndrome toxin. So not something to be taken lightly. These particular toxins can also be responsible for various forms of food poisoning. Okay? You typically hear about salmonella. Salmonella food poisoning is caused by the LPS layer of the gram-negative cell wall from the salmonella. These are gram-positive. So this is not a LPS toxin. This is a different kind of toxin that is actually secreted by the cells and can cause the symptoms of food poisoning. Streptococcus can cause a number of interesting diseases as well. Probably the most well-known one is strep throat. And you get, in this particular case, significant inflammation. You get this white, nasty-looking stuff in your pharyngeal region. Anytime you have inflammation of the throat, it is technically called pharyngitis. And pharyngitis, about 50% of the cases are viral, and the remainder are bacterial, including strep. That's really a challenge that doctors face, because if you take your kid, particularly, into the doctor with a horrible sore throat, and they're really miserable, you're going to want treatment, okay? And your immediate response is give them an antibiotic. But if it's a viral form of pharyngitis, it's not going to do any good. In the past, the only way a doctor could really determine whether it was strep or not was to take a throat culture, streak that out on some selective media, let it grow for a day or two before they could get the results and say, yes, it is strep. And then they could prescribe the antibiotic. Well, that's a couple-day waiting period. They've now developed rapid strep tests, which are antibody-based, and so you can go into the doctor, take a throat swab, and get a result in 30 minutes, and then the physician can make a good choice about whether to prescribe antibiotics or not. Anybody had a rapid strep test? Everybody probably have, yeah. One of the problems with not treating strep is that if it becomes systemic, if it gets into your blood and starts infecting other parts of the body, then you can get a real bad rash. That's called That's called scarlet fever. And if you get really, really serious infections, you can get heart damage. You know what that's called? It's called rheumatic fever. Okay, rheumatic fever is a secondary result of an untreated primary strep infection. And it's interesting what's happened. It's not the strep that is damaging your heart valves. What happens is your body starts reacting and producing antibodies against the strep bacteria. That's good. Okay. But those particular antigens that it's reacting to have partners or homologs, so you get a cross-reactivity between tissue and the heart valve, and the antibodies react to that as well. So you can get damage to the heart valves. They're showing you that a little bit over here as a result of untreated strep infection. So it's not something you want to take lightly. Strep infections can cause sinus infection, sinusitis. They can cause ear infections, otitis. And typically when the infection occurs, the body responds with an inflammatory response. And part of inflammation we'll see later is that fluid accumulates. And that can cause the pressure that's so painful in sinus infections and ear infections as well. But again, ear infections, probably about half of them are viral. So again, take your kid to the doctor with a horrible ear infection. It might be viral, it might be bacterial. You really have to culture the organism to know. One of the strep infections that got some real bad press, probably about 5-10 years ago, was this guy. This is necrotizing fasciitis. And this is a progressive disease, and it just destroys the tissues, and the skin sloughs off. It's a really nasty disease. You can see here that this particular person's leg has just been devastated by this. And it's primarily the result of toxins that are produced by these particular bacteria. So staph and strep we'll be coming back to a little bit more. Okay, volume 4. Phylum, Actinobacteria, a single class, Actinobacteria. We're not going to have much to do with these organisms. There are a couple of things I want to briefly mention, just because we may have instance to refer to them later on. Within that single class, is a genus called Actinomyces. These are sometimes referred to as the branched bacteria. These are kind of fungus-like in appearance. Did you look at any of your plates and look at the fungal colonies under the dissecting microscope at all? Fungal colonies usually look kind of fuzzy, and that's because they produce all these little filaments called hyphae, which grow and branch. Okay? Actinomyces do that too. They're very common in the soil. And if you ever pick up a handful of soil and smell it, it smells kind of musty. That's primarily the result of the presence of these actinomyces. They're very important decomposers in the soil, and they actually can, unlike us, decompose cellulose. And so plant cell walls can be broken down by enzymes that they produce. A second genus within this single class is the genus Streptomyces. And the only reason I really want to mention this right now is that this is one of the bacteria that was early identified as being able to produce an antibiotic. Anybody know what antibiotic this produces? Streptomycin. You may have heard of that before. We'll talk about the mode of action of streptomycin and what it does. And then finally, the third genus that's in the single class is the genus Mycobacterium. Word association. When you hear Mycobacterium, you should immediately think of something structural. You know what it is? These are organisms that are acid-fast. They're the ones that have that mycolic acid, that cord factor, impregnating their cell wall and gives them this characteristic of being acid-fast positive. Two species of the genus Mycobacterium are Mycobacterium tuberculosis and Mycobacterium lepri. So again, two important diseases. Here is a chest x-ray of a person with tuberculosis, and you can see these white shaded areas right here. Those are tubercles. That's where the name tuberculosis comes from. And when the bacteria infect the lungs, they tend to become encapsulated in this kind of granular material. And it's really hard for us right now in the age of antibiotics to appreciate what a scourge tuberculosis was in this country, particularly in the early 1900s. It was a huge, massive problem. And because of the lack of antibiotics, there really was very little course of treatment. Okay? Most people were isolated and put in sanatoriums. And a lot of times the idea was, since this was a respiratory disease, sometimes the sanatoriums were in the mountains. They were sanatoriums in Colorado. Sometimes the idea was is if you had them in the desert Southwest, that the result of the dry climate would make your lungs a little bit more easy to breathe. And really, until antibiotics were developed, tuberculosis was really a huge problem in this country. Unfortunately, tuberculosis is making a comeback because antibiotic-resistant strains of TB are now popping up. And that's particularly true in areas where there's a lot of poverty, sanitation isn't good, access to healthcare isn't good. And even if you do treat with antibiotics, the regimen for treatment with antibiotics for tuberculosis is very lengthy. You can be on antibiotics for a year or more to finally have to eradicate this particular disease. And it's hard to get people to stay with that. Leprosy, you probably more associate with biblical times. And again, people who suffered from leprosy were confined. They were forced to live in leper colonies. I think one of the last leper colonies that existed was on the island of Molokai in the Hawaiian Islands. And leprosy produces some very disfiguring and horrible outcomes. And it's pretty hard to hide leprosy infections. Okay? It tends to affect the face, the hands, the extremities. And part of the reason is that those are a little bit cooler regions of the body. Leprosy is very slow growing because it has a much lower temperature optimum. And the generation time, the time it takes for the cells to divide for leprosy, is like, 10 to 20 days as opposed to 10 to 20 minutes. What's really kind of interesting about this is because of that slow rate of growth, leprosy is really not that contagious. And so this practice of segregating people and putting them in leper colonies to protect yourself really wasn't based in very much really good science. We'll come back and talk about both of these diseases in a little bit more detail when we talk about kind of the medical microbiology aspect. And then finally, the last volume, this kind of catch-all volume that has several different phyla in it. There's only a couple of those particular 9 phyla that we're going to talk about at all. 2 to be exact. One of the phyla that we'll talk about in this particular volume is the phylum Chlamydia. And this is something you probably have heard about. Chlamydia is one of the kind of more common, unfortunately now, STDs. Chlamydia are characterized by being really, really small. This is the intestine of an individual suffering from a chlamydia infection. And these are the little microvilli, those little projections of the epithelial cells of the intestine. And here are the chlamydia. So I mean, they're not that much bigger than a single microvilli. These are also obligate intercellular parasites. In that way, they're similar to the rickettsia that we talked about earlier on. But they can only live and function inside of living cells. They're not transmitted by arthropods. These are primarily sexually transmitted diseases. They've got a pretty complicated life cycle. They actually have two morphological forms that differ during different stages of the life cycle. If we were going to talk about a medical microbiology course, we'd get into that life cycle, but don't have time. One of the things that can happen is that if a mother has an undiagnosed chlamydial infection, as the baby passes through the birth canal during the birthing process, the chlamydia can infect the baby, and one of the routes of entry that's most common is the eyes. And so this shows a young baby with an untreated ocular chlamydial infection, and that one's pretty nasty. So what they do now is after the baby's born, they treat the eyes so that if there is an undiagnosed infection, then it'll be taken care of. They used to use drops of silver nitrate. Have you ever gotten silver nitrate on your hands before in the chemistry lab? What's it do? Yeah, it turns your hands kind of brownish gray, and does it sting? Yeah, it burns. Okay, so putting those drops of silver nitrate in the eyes did kill these particular bacteria. Silver is a toxin, okay, but it hurt like heck. So now they're using more antibiotic eye drops now to replace the silver nitrate. I think they use erythromycin fairly commonly. Sometimes chlamydial infections really are fairly asymptomatic, and you really don't know you have them. There is some evidence that indicates that undiagnosed chlamydial infections can do things like scar fallopian tubes and might be responsible for some cases of infertility. And unless you're really looking for that, sometimes it's really difficult to say, why can't I conceive? And it might be the result of this undiagnosed chlamydial infection. And then the last phylum is the phylum Spirochetes. These are the spirochetes. You saw one in lab. You saw the Spirillum. Here are some spirochetes right here. They're characterized by that helical morphological arrangement. Some of the spirochetes have those axial filaments. I don't know if you remember that from cell structure, but the flagella wrap around the cell kind of like a belt. And as it contracts, the cell corkscrews and burrows into tissues. A particularly important genus within this particular phylum is the genus Treponema and the species Palladium. And this is a causative agent of syphilis. So syphilis is a spirochete. Syphilis was another scourge of the pre-antibiotic year. And people were desperate to try to figure out a way to treat syphilis because it was a very common sexually transmitted disease. We're going to talk about a little bit later on kind of the history of the development of medical treatments to try to kill bacteria. And some pretty toxic brews were made, pre-antibiotics, that were intended to try to kill pathogens like the syphilis pathogen. One of the early pioneers was a microbiologist physician by the name of Ehrlich, Paul Ehrlich. And his goal in life was to develop a drug that he called a magic bullet. And the idea of the magic bullet was that it would kill the pathogen but not harm the host. So his life work was kind of geared toward finding a magic bullet to treat syphilis. And he developed a drug to treat syphilis, and he called it salvarsan. The salve part was that it was intended to bring salvation or relief from this particular disease. The basis of this compound, ARS, was arsenic. So this was an arsenic-based compound. Well, arsenic is toxic to syphilis, but it's toxic to human cells as well. So this wasn't a very selective magic bullet. And consequently, there were a lot of toxic side effects. Many people argued the drug was worse than the disease. But by the 50s, and particularly when World War II was ending and a lot of soldiers were coming back with STDs, antibiotics cleared this up immediately. Tetracycline, the wonder drug, one of the antibiotics we'll talk about. Take a few pills and you're done. And so that really did result in some really kind of loosening of public health standards and concerns. Prevention of these diseases wasn't so much of an issue now, just treated after the fact. But again, now we're coming out with drug-resistant forms of some of these bacteria, like the Treponema bacteria. And then finally, the second genus, the genus Borrelia, species Burgdorfii. Whoops, forgot to show you a couple things, a couple of images. Syphilis goes through usually a set of 3 stages, depending upon whether it's treated or not. This represents primary syphilis, and these little lesions that can form on the genitalia or the lips are called chancres, and that's the indicator of the first infection. But you can recover from that, and the lesions can disappear, but you can still harbor the syphilis. And it can break out later on again into a secondary infection, which is sometimes the result of a rash. And so this is a secondary syphilitic rash. That can even disappear, and you can still harbor the pathogen, and then it can start eroding away tissues, and that is tertiary syphilis. And you can see here this person's nose is just being kind of disintegrated away as a result of untreated tertiary syphilis. And tertiary syphilis is lethal. I think Al Capone, the notorious gangster, died from tertiary syphilis. Okay? So again, a disease not to be taken lightly, and just because the original symptoms disappear doesn't mean you're still not infected. Antibiotic treatment is very effective, though, at killing that. This is untreated situations. Okay, so here is Borrelia burgdorfii. This is the causative agent, right here, a spirochete. This is the vector that transmits that particular agent from white-tailed deer primarily to humans. And this is a tick. And this is one of the characteristic manifestations of this particular disease. You know the disease we're talking about? This is Lyme disease, okay? And that bullet rash that you see right here, which isn't always as pronounced as this picture shows, is one of the characteristics. There's a lot of other symptoms as well. There can be incredible joint pain, there can be real fatigue, and this is a disease that is really very difficult to diagnose. But again, if you nail it down, it is treatable by antibiotics. Lyme disease used to be fairly endemic to just kind of the northeastern part of the United States. And that's because its host, the white-tailed deer, was primarily restricted to that area. But as climate has changed, the range of the white-tailed deer has expanded. And it's in Indiana now, for example. And anytime that host is present and the vector, the tick, is present, then you've got the risk of potentially transmitting this disease from one animal to another. And so again, this is another reason why when you go out in the woods, you really should be careful about whether you picked up any ticks, because they are fairly nasty vectors for some arthropod-borne diseases. Okay, so the bottom line, what do I have to know? Okay, as far as classification is concerned. I think it's important kind of historically to know the name of the 4 divisions in Berge's first edition of systematics. So the Gracilicutes, the Firmicutes, the Tendericides, the Melicides, because you hear some of those names again. Okay? What do you need to know about the second edition, the one we've been going through? You need to know the names of the 5 volumes. And for that last volume that has all those lists of things, you can know a couple of them. Okay. Which domains are included in those? That's pretty easy. It's only the first volume that has 2 domains. The rest of them are just 1 domain. The number of phyla and classes in each of those volumes. Many times there's only a single phylum, a single class. Sometimes there are 9 phyla but no classes. But just get an idea of how those things are kind of organized. And then for representative organisms we've talked about, what's the organism? What volume would it be in? And then what phylum and class or genus, if I told you that. Okay? And this is on your PowerPoint slides, so you don't have to try to write all this down. But this will give you a way to kind of make some sense about what are my expectations going to be in terms of how much of this taxonomy you'll have to know. So Tuesday, we will pick up with the growth. And theoretically, we might even get through that material that would be the stopping point for the exam on Tuesday, which would be nice. So Monday, come in and inoculate your media, and you'll be good to go. See you Tuesday.